Okay, so chapter 10, we're going to be looking at the molecular biology of the gene. So what we're going to be talking about is we just went over mitosis and meiosis, and now we're going to talk about how DNA is turned into a protein. So if I can get my lucky pen to work. So here are the big ideas. What is the structure of DNA? How does it replicate? How does it get translated? And then we're going to go a little bit into viruses and bacteria. So the structure of DNA. So before DNA was discovered, they used to think proteins were the genetic material of the cell. That was the proteins that were passed down. And proteins are made from 20 different amino acids, and DNA was only to have 4 kinds of nucleotides. So this kind of made a little bit more sense to people. So when they started studying bacteria and viruses, it kind of brought in that field of molecular biology and looking at heredity at the very molecular level. And when we did that, we kind of found the role of DNA in heredity. In 1928, Mr. Frederick Griffith, he discovered that there's a transforming factor that you could take and transfer into a bacterial cell. So when he took a pathogen that they had heated up and killed, and to put it, expose it to harmless bacteria, some of the harmless bacteria were converted to disease-causing bacteria. So how did these heat-killed bacteria turn these harmless bacteria into disease-causing bacteria? And so the disease-causing characteristic was inherited by those descendants of the transformed cells. So they were able to pass it on. So he kind of showed that there was some sort of heredity going on, some sort of change that occurred, transfer that occurred from the heat-killed bacteria to the regular bacteria that then became hereditary. So in 1952, Alfred Hershey and Martha Chase, they used bacteriophages, and these are really cool, and I'll show you pictures, to show that DNA is the genetic material. And so they're looking at a virus that infects E. coli, right? So bacteriophage, or phages, right, or viruses that infect bacteria, phages were labeled with radioactive sulfur to detect proteins or to detect DNA. So the bacteria were infected with either type of label phage and tried to determine where did that radioactive material go and what happened to it. So what they found, that the sulfur-labeled proteins stayed with the phages outside, but the DNA went inside, and then that got transferred. So it was a pretty neat experiment. So these are bacteriophages, and these guys are awesome because they look like alien spaceships, right? And essentially they're just protein and DNA. That's essentially all they are. And so if you look at those legs, right, they connect with antigens on the cell of the bacteria, and then they inject their DNA inside the bacteria. And then that does its trick, and the bacteria starts forming more and more and more of them. So first they put the radioactive protein, right, and they infected the bacteria with this, right, and then they went through the centrifuge, and the radioactive was in the liquid where the DNA, the empty protein shell, remained. But then in batch 2, when they did the radioactive DNA and then infected the bacteria, did the centrifuge, then the DNA was actually inside of the bacteria itself. So This is kind of how the phage works. It attaches itself, it injects the DNA. The phage DNA directs the host cell to make all sorts of other phages and put them all together. And then once they're put all together, there's so many of them, they break out, and it kills the bacteria cell, and then there's a whole bunch of more little viruses cruising around. So DNA and RNA. DNA, deoxyribonucleic acid, RNA, ribonucleic acid. Both of them are those nucleic acids, and that's what the Na stands for. So when you see DNA, RNA, that's kind of what you should think about. So one of the two strands of DNA is a polynucleotide. So it's a polymer, multiple nucleotides. So it's a chain. So a nucleotide is composed of nitrogenous base, 5-carbon sugar, and a phosphate group. And that so you have phosphate, sugar, nitrogenous base. And it's the sugar phosphate backbone that connects that DNA. So when we talk about nucleotides, they're nitrogen-containing bases, and we just label them A, C, T, and G. But the correct word for them is adenine, cytosine, thymine, and guanine. So DNA is a double helix, and this will show you a picture a little bit later, but it's kind of twisted around. So you have that sugar phosphate backbone, and on the right of your screen you can kind of see the phosphate group and the sugar group and the nitrogenous base, and it's those bases that connect with other bases through. And it's a sugar phosphate backbone that kind of brings it into a long strand. So you have pyrimidines, which are thymine and cytosine, and purines, which are adenine and guanine. So RNA is a little different. It uses the sugar ribose instead of deoxyribose, right? So it has an oxygen there, and because of that, it uses the nitrogenous base uracil instead of thymine. So instead of T, it's a U. So if you see a strand and it has a U and no T's, you know it's an RNA and not a DNA. And so here's kind of your computer modeled where you look at the ribose and the phosphates and the bases, and you have uracil up there instead of thymine. So after the Hershey-Chase experiment in 1952, after they demonstrated that the genetic material was most likely DNA, there was this huge race to see who could describe the structure, right? Who could lay out this bag of snakes and explain how the heredity of life occurs and how we can inherit different ideas. So Watson Crick, most people know them. They deduced the secondary structure of DNA, but they used work from Rosalind Franklin. Unfortunately, because Rosalind was using X-rays, and they didn't know that it was potentially dangerous, she died very, very young. And there is a bit of controversy that Watson and Crick did not have permission to take her work, and they just kind of walked in and took their work. And at that time, it was a bit misogynistic in the science world about that females can't be scientists, and so a lot of males took advantage of work from females and didn't give them credit. And I think it's pretty obvious that Watson and Crick was one of those. And they even admitted to it later on in life, but after she was dead. So they took the data and they found, Chargaffs found that the amount of adenine was equal to the amount of thymine, and the amount of guanine was equal to that of cytosine. So they knew that G, T, A, and G, C, and A and T kind of matched one another. There's Rosalind in one of her X-rays of the DNA that was very influential for Watson and Crick. to deduce the structure of DNA. And so Watson Crick reported that DNA consisted of 2 polynucleotide strands wrapped into a double helix. And so you have the sugar phosphate backbone. The nitrogenous bases are perpendicular to the backbone, and specific pairs would give a uniform shape, A with T, G with C. And notice they form with hydrogen bonds. So there's a famous picture of Watson and Crick explaining the structure of DNA. So this is how you can kind of think of it. If you take a ladder and you twist it, right, that's how you get kind of your double helix cruising around. And it also saves space, because in every cell in our body there's 3 feet of DNA, and our cells are really, really small. So there's lots of different ways that the body kind of takes the DNA and twists it and wraps it and to make a lot of space. So on the left you have the ribbon model, which shows the base pairs and it's twisting around, and then you have kind of your partial chemical structure, and you see between the C and G's there's 3 hydrogen bonds, and A and T's there's 2 hydrogen bonds. And then on the right, your computer model that kind of shows the space filling model. So 1962, the Nobel Prize was awarded to Watson and Crick. And Rosalind Franklin probably would have received the prize, but she died from cancer in 1958, and Nobel Prizes are not awarded posthumously, meaning after death. Right. So the Watson-Crick model was really important because now we kind of understood genes and chromosomes a lot better, and how genetic information is, how that recipe book is enclosed in there. So replication, how do we read that recipe book? Right, we got our chocolate chip cookie recipe. How do we translate it? What the heck is 1 tablespoon? That kind of thing. So when they described the DNA, the Watson and Crick showed that the structure of DNA, there was a possible copying mechanism. And in fact, it's a DNA replication is a semi-conservative model. So what happens, the 2 strands separate, and each strand is used as a pattern to produce another strand. And so each new DNA helix has 1 old strand and 1 new strand. So that's what it means by semi-conservative. You get 1 new, 1 old. So here's a parental molecule, and that splits up, and then your nucleotides, the ones in the in your cell, just come in and they match up because of chemistry. I'm not going to get too much into the biochemical mechanisms. Just know that there's chemical attractions that makes it work, and then we have 2 identical molecules, both with 1 old and 1 new. And so here's a double helix demonstration. You have your daughter molecules and the parent molecules, and it splits. Those hydrogen bonds split, and new ones are formed. So replication for DNA is going to go in 2 different directions. And so when we look at it splitting, one will go up and one will go kind of in, one will go continuously, one will go in kind of chunks, and we'll talk about this in a second. So DNA unwinds at an origin. It's got to make a little bubble. So if you think of just unwinding the ladder at one specific location, that's a bubble. And then replication goes in both directions, and it ends when products from the bubbles merge with each other. Okay, so DNA replication occurs in the 5 to 3, the 5' to 3' direction, and so when you replicate it, it's continuous on the 3' to 5' template. But it's discontinuous in they call them Mosaic fragments. And I don't think your book discusses that. But on the 5' to 3' strand, it has to do short segments. So the proteins involved are ligase, which brings the small fragments into a continuous chain. And polymerase adds nucleotides, proofreads, and corrects. And there's all sorts of animations for your book and online and Mastering Biology that you can look at to help understand this as you move through. So polymerase and ligase, when we're discussing DNA, of course, they can repair damage in DNA that's been damaged by radiation, toxic chemicals, and things like that. Of course, it's not perfect, and if there's a ton of damage, then it can't repair all of it. And so the replication makes sure that all the body cells, the somatic cells, and multicellular organisms have the same genetic information. So the cells in your eyes have the same genetic information as the cells in your toes. Okay, they just don't produce proteins for toes in your eyes, right? And I think we talked about that earlier. So here are your bubbles. Your parental strand, they bubble and they replicate, and you see the purple elongating, and then we get two daughter strands of molecules. So this, in the synthesis phase of interphase, right, that's when that happens. So here we go, the 5 to 3 end, and what they mean, and this is a little more clear, so the 5' to 3'. So if that was confusing to you, hopefully this is going to make it a little better. And so what they're talking about is the carbon. And the way, this is a carbon here, this is a carbon here, here, here. And so we have this 5' carbon. Okay, and that's just simply the way they count it in chemistry. Just make sure you kind of understand the basics. I'm not going to get into the biochemistry. So we got a 5' carbon, and then it moves from the 5 to 3 direction. So wherever that 5' is at the top and the 3' is at the bottom, and here we have the 3' end, and so you see the carbon here, 1, 2, 3, 4, and 5. So if you notice, it's flat here. Oh, there goes my pen. Dang it. And it's flat up here, right? So that pentose is reversed. That 5' sugar is reversed. And so there's your change. And hopefully that kind of explains a little more what I mean by 5' to 3' and 3' to 5'. Okay. So we got our polymerase, and those are proteins. And if you notice when it's going from 3 to 5, everything is hunky-dory. So the daughter strand here is synthesized continuously. And that's all fine and happy. But on the other end, it is not. And so it has to do it in fragments. And in fact, it'll come and do this and then circle back and forth and back and forth. And this is very important because our DNA is linear. So if we look at a chromosome here, and it's splitting, and one strand is continuous, so this strand is continuous, it's made, and the other one is not. It's done in loops and fragments coming back and forth and back and forth. When we get down here at the bottom, there's a little chunk that cannot be replicated. And so that little chunk gets cut off. Okay? And so when we look at chromosomes in general, they have at the end, let me change colors and see if this is a little more helpful. They have at the ends an enzyme, which is called telomerase. Tele means distance, long way, telomerase. And so when it's replicating and things get cut off here, 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 it can replicate a whole bunch of times without ever actually getting into the actual alleles that code. They think part of aging is when you run out of telomerase, and you start getting actual genes and alleles cut off, so you're not making proteins. They've tested this by adding telomerase to the chromosomes of fruit flies, and took a fruit fly that should last a couple days, and it's lived for a couple months. The problem is it didn't live very well. It wasn't a good quality of life. So that's why it's kind of important to understand the two different directions of DNA, because we have linear chromosomes. When we talk about bacteria, they have a circular chromosome, so there is no issue. They don't need any telomerase. Okay, the flow of genetic formation from DNA to RNA to protein. All right, let's talk a little bit about this. The gene to protein dogma. So supposedly our DNA, our recipe book codes for chocolate chip cookies. And then that gets transcribed into RNA. RNA gets translated into the actual proteins or the cookies, right? And then those proteins make us up. But it's not entirely accurate all of the time. Okay, the gene to protein dogma is correct most of the time, but sometimes our body can, let's say we have coding for blue eyes. Sometimes our body can switch that out and even have brown eyes when our DNA says we should have blue eyes. So just be aware of that your body can alter the proteins being made, even though your DNA specifies for something. But let's look at the gene to protein dogma. So DNA specifies traits by dictating proteins. And then we have a molecular chain of command, right? So you have kind of the guys in the office, which are in the nucleus. That's the DNA, and they have the blueprints for the house you're building or whatever it is. And then you have RNA that's also in the nucleus, but it's also in the cytoplasm, and that's where it's formed into a protein. So RNA is kind of the engineer that goes to the office, picks up the blueprints, and takes them to the job site. And then the job site, they're translated, and we build a house. So transcription, trans meaning movement, script meaning write, writing down, a scribe. So transcription is the synthesis of RNA under the direction of DNA. Translation, once again, trans, movement. And so we have, oh, there goes my pen. Translation is when we take the RNA and try and make sense of it. We translate the language of the DNA into a protein that will function. So here's your graphic on that. We have DNA, the double helix, and that gets transcribed, and so you have this RNA molecule, and that leaves the nucleus. So remember, RNA can be in the nucleus or in the cytoplasm. DNA is not in the cytoplasm. And then that gets translated into a protein. Remember our ribosomes, that's where the translation takes place. So we have this connection between gene and protein. And so they initially thought one gene, one enzyme, and that was based on inherited metabolic diseases. And then that was expanded to include all proteins. And then most recently they go to the one gene, one polypeptide hypothesis, which recognizes that some proteins are composed of multi-polypeptides. So some proteins come together, multiple proteins can come together and things like that, and change how your phenotype, how things are expressed. And so we have kind of our nucleus there. And so the sequence of nucleotides in DNA, it has that code for constructing the protein or the house. When we're building a house, we have to convert because the blueprints to what the house should be. So if you notice, blueprints obviously aren't to scale, so you have to measure the length of the wall in the blueprint and translate that to what it's supposed to be for the house. Um, Transcription can rewrite the DNA code into RNA and uses the same language from the same nucleotides. And then what they do with that information from the gene to protein is based on a triplet code. They are called codons. Right? So 3 of those nucleotides together code for certain polypeptides. And so this is the language, this is the translation. And so when we look at each amino acid, there's 64 codons possible from those 4 nucleotides. So when we're dealing with 3, an arrangement of 3. And so there is some amino acids have more, so there is some layover. So here we are, the DNA molecule, and there's a certain gene 1 and gene 2 and gene 3. And then that gets transcribed in RNA and then translated into polypeptide using codons of 3. So in this case, it made it easy and said AAA and CCG and so on and so forth. So 3 nucleotides are going to specify 1 amino acid. That is the codon. 61 codons correspond to amino acids. So that means there are signals, codon signals, to start and stop transcription. So there's 3 of those. There were 64 total codons, but 3 of them stop. So it tells your body, okay, stop making the protein right here. There's redundancy, so more than 1 codon for some amino acids, because if you remember, there's 20 amino acids, right? It's unambiguous in that any codon for 1 amino acid does not code for any other amino acid, and it's almost universal, meaning it's shared by organisms from the simplest bacteria to the most complex plants and animals. There is no punctuation in codons. They're adjacent to one another. There's no gaps in between codons. It's just reading them by 3. And so here you go. Here's the code. Each one of those, if you look at the left, there's U, C, A, and G. We're not T, C, A, and G because we're talking about RNA. Right? And so if the first one on the left is a U, and then the middle one, if you look on the top, U, C, A, and G is a U, and then on the right, it's U, C, A, and G. So you can kind of follow your codons. And if you remember, there's 3 stop codons, and there's 1 start. The AUG is a start. UAA, UAG, UGA, those are stop codons. And then as you go through, you can see all those different amino acids. So here's kind of a blow up of that graphic we saw before. And what you can do sometimes is make mice glow in the dark. We can make us glow in the dark if you wanted to by adding phosphorescent alleles into or proteins into our skin cells. I did this with fish as an undergraduate. We made fish glow under a black light. It was kind of fun. So, transcription. An RNA molecule is transcribed from a DNA template, and that's very similar to when DNA replicates. It's kind of the same process, but instead we're making RNA. RNA nucleotides are linked by transcription enzymes, which is RNA polymerase. So ACE enzyme, right? It helps things move. It's a protein that kind of helps this process go along. And then we have specific sequences of nucleotides along the DNA mark where to stop and where to start, where to start, where to stop, back and forth. The start transcribing signal is a sequence called a promoter. It promotes transcription. And so transcription starts with the initiation when RNA attaches to that promoter region. And during the elongation phase, the RNA grows longer and longer and longer, and then as it peels away, the DNA rejoins. So if you think of that bubble, like we looked up before, instead of the whole chromosome splitting apart and replicating, just a small little bit where the allele is, there's a little bubble, opens up, RNA comes in, starts to elongate, and then once it peels away, DNA will rejoin. And so in the third phase of termination, the RNA polymerase reaches a sequence called a terminator, which just says stop right here. So you have the promoter and the terminator and elongation. Promoter is first phase, elongation second phase, termination is third phase. Okay, and here you go. So here's your polymerase, it goes to the DNA, there's a little bubble made, and then the nucleotides come in and starts to make an RNA and transcribes it, and when it's done, the DNA comes back together. So initiation, elongation, and termination. So that RNA polymerase will attach to the DNA where that promoter region is. And so the polymerase won't attach where there isn't a promoter. But it'll start to split the double helix apart and RNA will form, and then once it gets to the terminator, it pops off the chromosome. mRNA, messenger RNA, right? There's lots of different types of RNA, but mRNA, messenger RNA, encodes amino acid sequences, and then it takes that genetic message into the cell. And so in eukaryotes, so what our RNA has to exit through those pores. But in prokaryotes, the RNA can be done right there because there is no nucleus, right? And so our mRNA has introns and exons. Introns interrupt the sequences, and exons are the coding regions. And so eukaryotic mRNA undergoes processing. It has to be processed before. All sorts of different things, very complex when you start getting to eukaryotes. So RNA splicing takes out the introns, joins exons. Okay. There's a cap and a tail, and this facilitates the export of mRNA from the nucleus and protects it from cellular enzymes because your cells will attack foreign DNA if it's in your body. Because it doesn't want to use all its energy to make stuff for something that's not you. So that's why our mRNA has to be protected with those caps. So here's your graphic. Exons, introns, pop out the introns and have the exons, put the caps on, and then it can leave the nucleus. tRNA, it's a language interpreter. It figures out what that codon is and then what specific amino acid it's supposed to go to. So, and they do that by grabbing the appropriate amino acid and looking for, they have what's called an anticodon. So you have codons and anticodons. Anticodons are on the tRNA, codons are found in the mRNA. Here's your graphic. So it's got that amino acid attachment site, and it's got the anticodon. And that anticodon only fits certain codons in the mRNA. And so obviously this takes energies, and that's why you have your ATP all through there. And so translation occurs on the surface of the ribosome. Ribosomes coordinate the function of mRNA and tRNA, and then so they're kind of the job site manager that's building that house, and so they coordinate all those different things. And there's two subunits to the ribosomes. Think of it like the manager and assistant manager, I guess. So in each subunit has ribosomal RNAs and proteins. So each ribosomes are made up of RNA and proteins themselves. and the subunits come together, and ribosomes have the binding sites. And so they're cruising around, and then they come together around an mRNA, and they start bringing in tRNA, which starts to make polypeptides, and which makes the protein. So there's your large subunit and your small subunit and your TNA binding sites. Okay, so there's your mRNA moving through there, and a bunch of tRNAs coming in and out all the time, and making a growing polypeptide chain. And so we can divide this into 3 phases: initiation, elongation, and termination. And then the initiation brings together mRNA, and then a tRNA has the first amino acid and the 2 subunits of the ribosome. That starts the initiation. And then when more tRNA comes in, that continues, and it kind of builds from there. So initiation has 2 steps. mRNA molecules bind to the small ribosomal unit, and the first tRNA binds to the mRNA at the start codon, and that's the AUG codon. And then the first tRNA anticodon is UAC, which is just the exact opposite of what nucleotides of what would fit into a messenger RNA. So if we look at the anticodon, you should be able to move it back to the DNA. If we give you DNA, you should be able to translate it and move it through to the mRNA and the tRNA codons. The large ribosomal subunit joins the small subunit, allowing the ribosome to function. So it won't function unless it has both units, and they kind of sandwich that mRNA in between them. First tRNA has a P site, and then the A site can take the next tRNA. And so you have your caps, your start of the genetic message, the end, and another cap. And so here's your first small unit, the mRNA, the anticodon on the tRNA, and your large ribosomal unit. So once the initiation is complete, amino acids are added one by one to make the first amino acid. And that's elongation, is when we start adding more and more amino acids. So codon recognition. The anticodon on the tRNA that has the amino acid with it pairs with the mRNA, and so through the chemical reaction, when the anticodon matches up, the bond between the tRNA and the peptide is broken, and it's formed onto the chain. So the translocation, or tRNA, is released from the P site, and the ribosome moves the tRNA A site to P site. So that continues until it gets to the termination, or the stop codon. And then once it gets to the stop codons, that ribosome breaks apart and kind of goes its own separate way. So here's a circle: codon recognition, polypeptide bond formation, translocation, and so it'll come together and then pop off once it's done. So transcription is synthesis, transcribed, right, of RNA from DNA. Translation is telling what protein it means. Transcription is in the nucleus. That's a great test question that is often asked. And mRNA has to go from the nucleus to the cytoplasm. And so when we talk about translation, not transcription, translation, it's 4 steps. And they all occur in the cytoplasm. Another great test question. So you have the amino acid attachment, initiation of the polypeptide, the elongation, and termination. Right? So transcription inside the nucleus, and when we get to the cytoplasm, we have the amino acid attachment on the tRNA. We have the ribosomal units coming together over the mRNA, starting to create the growing polypeptide as it moves through, and then once it gets to termination, they all kind of go their separate ways. And this all happens. It's this is amazing because there is so much chemistry going on in here. It all happens in a split second. It really happens very, very, very quickly. So you would think it would take a long time, but it doesn't. Happens very rapidly. So mutations, right? They can change the sequence of DNA, which changes the protein that is made. Proteins can change phenotype. So they can be chromosomal regions or just a single nucleotide pair. So base mutation is kind of a replacement of one nucleotide to another. And it may have no effect at all, because if you remember looking at the codon sequence, that some codons, multiple codons code for the same amino acid. So if there's a base substitution, but it may not change what protein's made. And so that's called a silent mutation. When we change the amino acid coding, producing a missense mutation, it creates a different amino acid. And base substitution can also lead or produce an improved protein that enhances the success of a mutant organism. That doesn't happen very often, but it does happen. So a lot of people think mutations are all going to be bad but they're not always bad, and occasionally they are very helpful. Sometimes we get lots of mutations and they don't really change anything because of the different codons coding for the same amino acid. So they can change an amino acid into a stop codon, producing a nonsense mutation. And so we also have deletions. So if you delete, it can change the entire sequence after that and produce either a functional or non-functional protein. So mutagenesis, genesis, creation, mutation, so it's the production of mutations. They can be spontaneous errors. They can happen by chemicals. They can happen by X-rays, ultraviolet light, high energy radiation. All sorts of different things can create mutagens. So here we are with normal hemoglobin DNA that will produce your amino acid, and then you have the mutant hemoglobin DNA, something just slightly different, and that produces sickle cell hemoglobin. And this is very interesting because just one nucleotide mutation can confer a advantageous trait for people in high malaria areas, or a non-advantageous trait for people not in high malaria areas. So just one mutation can confer a benefit. Sickle cell is where your blood forms kind of this sickle shape, and in malaria areas it helps prevent malaria from occurring in the body. So people that are heterozygotes, or meaning they have some normal cells and some mutant cells, have an evolutionary advantage over those that have normal blood. But if you notice, there's some football players that when they go to play at Denver at Mile High Stadium, they can't play because they have sickle cell, and it would kill them to play that high because there's not enough oxygen. They have to get more oxygen than others. So it can be a detriment or a benefit depending on the environment. So here's another example: normal gene AUG, start codon. And then if we look at a nucleotide substitution or a nucleotide deletion, what can happen, or a nucleotide insertion, we can get different proteins. So viruses and bacteria. Viruses are, they call them genes in a box, and that's, it's pretty much just DNA wrapped in a protein coat, and they call that a capsid. And they have two types of reproductive cycles, viruses: lytic cycles, where particles are produced using host cell components, and then the host cell lyses and viruses are released. And the lysogenic, where viral DNA is inserted into the host chromosome by recombination, and then the viral DNA is duplicated along with the host chromosome during each cell division. The inserted phage DNA is called prophage. And so viruses are really interesting. So if we look, here's the phage, it attaches to the bacteria, inserts that bacteria. All of a sudden, the cell has that phage DNA in it, and it starts to create all the different parts. It starts to assemble the parts, and pretty soon there's so much of it, they bust out of the cell, killing the bacteria, and there's a lot more viruses cruising around. So the lysogenic cycle, okay? So the phage comes in, inserts its bacteria, and then it starts to gets recombined into the bacterial cell DNA, and so every time it splits, that virus DNA is in there, and at any time could be produced to kill the bacteria. Canker sores are kind of like this. So sometimes you'll get canker sores and sometimes you won't. But that viral bacteria has been inserted into your cell's DNA, and every time your cell reproduces, it carries it with it. And then something starts it and causes it to pop out. So there's the short and there's the long cycle. So viruses can disease animals, plants, and the DNA or RNA in a virus can cause those disease in animals. So the typical animal virus has a membrane outer envelope and projecting spikes of glycoprotein. Those spikes kind of feel for the right cell, so they infect the correct cell. And so that envelope helps the virus enter, leaves the host cell. And then many animal viruses have RNA rather than DNA as their genetic material. And that's cold, measles, mumps, polio, AIDS, or HIV. And so we have these reproductive cycles of the mumps virus, which is a typical enveloped RNA virus, and it's got 7 steps. Entry of the protein, RNA into the cell, uncoating, the removal of the protein coat, RNA synthesis, mRNA synthesizes using the viral enzyme, protein synthesis, mRNA is used to make viral proteins, the new viral genome production, mRNA is used as a template to synthesize new viral genomes, then it's assembled, the new proteins assemble around the new viral RNA, and then they exit, and they cloak themselves in the host cell's plasma membrane. So the herpes virus sometimes reproduces in the cell's nucleus. Most plant viruses are RNA viruses. To infect a plant, they have to get past the outer protective layer of the plant. And then they spread from cell to cell through the plasmodesmata, which are those holes in between the cells. And they can spread to other plants by insects, herbivores, humans, farming tools. And there's really not a whole lot of cures for a lot of the viral diseases. That's why if you go to the doctor and it's a virus, he just says, well, you have to let it run its course, because we can't really do much. Antivirals are pretty nasty. So here's your graphic. The spikes on that viral genome allows it entry into the cell. All of a sudden now there's some RNA in the cell. That protein comes apart, and then there's ribosomes and nucleotides everywhere in the cell, and so RNA gets synthesized, and the RNA makes proteins, and then those proteins help form and assemble, and then it pops out using the cell membrane to kind of cloak it so your immune system doesn't find it and kill it. So emerging viruses, they have a sudden onset and they're kind of hard to deal with. These are the AIDS, Ebola, West Nile, SARS. These are very, very difficult to deal with. So three processes contribute to the emergence of viral diseases. One is mutation. RNA viruses can mutate very rapidly, so if your body says, all right, this specific RNA is bad, and goes out and attacks it, it can mutate to the point where very quickly to where there's many viruses still hanging around and your body hasn't made enough killer cells to go find them. AIDS, HIV does this very, very well. I think it's somewhere around might be 50% mutation rate. It's just incredibly high. So they spread through contact between species. For example, HIV did not, some people say, well, HIV did not occur by humans mating with monkeys. It came by humans eating monkeys. So the butchering of bushmeat, right, is how the SIV mutated into HIV, and then spreads from human to human. And in fact, the highest people at risk for contacting HIV are females, particularly married females. This idea that those who get HIV had a promiscuous life is false. It's really false. Just because someone has HIV does not mean they had a promiscuous life. And in fact, if you look at worldwide, there's kind of an equal number of males and females, and in many places females are at higher risk for HIV when they're married, and that's the only person they have any sexual contact with is their husband. There's a lot of misconceptions about HIV in the U.S. So it can spread from isolated human populations to larger human populations. And with our travel, airlines, boats, how fast we travel from one place to another, emerging diseases are nigh to impossible to try to contain. If it's airborne virus, a lot of people start wearing masks and things like that to help protect themselves against the virus. So HIV, acquired immunodeficiency syndrome. It's an RNA virus, has 2 copies of its RNA, and carries molecules of reverse transcriptase. This is which causes reverse transcription, producing DNA from an RNA template. So instead of going from DNA to RNA, it can go from RNA to DNA. It has a high mutation rate. So here's an HIV virus. You have your envelope, your glycoprotein coat, 2 strands, identical strands of RNA, and reverse transcriptase, 2 copies. That's all it is. That's it right there. And so after it's encoded in the cytoplasm, it makes a DNA strand from RNA, and then adds a complementary DNA strand. And so now we have a double-stranded viral DNA entering the nucleus and integrates into the chromosomes, becoming a provirus, which then used to make mRNA, which then translated to make viral proteins, and then they're assembled, leave the host cell, and can infect other cells. And there's all sorts of tricky things about HIV. Make it very, very difficult to deal with. So viral RNA and your reverse transcriptase makes the DNA, and then makes parts of the cell, and then it buds out and goes and infects other cells. So some agents are made only of RNA or protein. So viroids are small circular RNA molecules that can infect plants. So there isn't even a coating on these. It's just simply a circular RNA molecule. And prions are just proteins. That's all they are. Yet we can get mad cow disease. We can die if we eat some of these. So these are incredibly simple but very dangerous aspects of our environment. So viral reproduction allows researchers to learn more about the mechanisms that regulate DNA replication. So it we can use viruses to study how DNA works and how gene expression works. We've done very well with this. So, and bacteria are valuable, but for different reasons. Bacterial DNA is a single closed-loop chromosome. It's that circular chromosome like I showed you. And then bacteria cells divide and replicate by binary fission. And binary fission is an asexual process. And so bacteria in a colony are genetically identical to the parent cell. And so we can use this to kind of understand the process of gene to protein. And so bacteria use three mechanisms to move genes from cell to cell. So to create genetic variation. Transformation, where it uptakes DNA from the environment. Transduction is transferred by phages, and conjugation is kind of like this mating through a sex pili. Once new DNA gets into the bacterial cell, parts of it may then integrate into the recipient's chromosome. So we use this all the time to do things like create insulin. We can use bacteria to create insulin because we can put the DNA into the bacteria, and then the bacteria will do the work of forming the insulin for us. So here we are, a fragment of DNA from another cell enters the bacteria cell, or a phage will inject it, or there's sex pili or mating bridge, right? And so you have a donor cell and a recipient cell. And then you have crossovers and degraded DNA, recombinant chromosomes, and different things like that will change proteins made by that bacteria. So they call this F factor, not X factor, F factor, and that's the ability of the E. coli cell to carry out the conjugation. During conjugation, the F factor is integrated into the bacterial chromosomes, and then the donor chromosome starts replicating at that origin of replication, and then we have a growing copy of the DNA peels off and heads into the recipient cell. And it serves as the leading end of the transferred DNA. So here's your graphic. F factor, integrated DNA, and then that the F factor starts to replicate and transfer, and then only part of the chromosome transfers, and then recombination can occur, and so we can have this plasmid, and then F factor replicates and then can be transferred, and then the new cell is now a donor cell. So here's that was kind of the a blow up of the other slide. So F factors can exist as a plasmid. Plasmids are just small circular DNA molecules. They're not the regular DNA, they're just small circular DNA molecules that kind of can still create proteins. Our plasmids pose serious problems for human medicine because they carry genes for enzymes that destroy antibiotics. So this is kind of the basic for the mechanism for why antibiotics are no longer as effective as they were. So in here's a bacterial cell with DNA, all the DNA spread out, and if you see these small circular ones, those are plasmids. Okay, so that is chapter 10. Wow, I went through it super fast, and I did that to try to keep this under an hour. But go ahead and take good notes on it, and this should be the end of the module. In our next module, we're going to talk about evolution.